Hi, everyone. Welcome to the second part of Chapter 8. A few announcements that I want to make as we come to the close of our first in-person week of classes. So if you're watching this in the fall of 2020, I'm recording this on September the 11th, and I wanted to just mention a few things. First of all, what I have discovered is that doing the in-person review sessions or the classes, the face-to-face classes, it's going to be difficult for me to do Zoom recordings. So there will be no Zoom recordings. Of the in-class sessions. Now remember, the in-class sessions do not present new material. The only thing that I do in the face-to-face sessions is answer questions and review these lecture recordings. So if you're watching these lecture recordings from home and you want to remain online, it is okay to treat this as an online course, okay? You do not have to come to class if you are nervous about your possible virus exposure or if you're feeling ill, okay? So again, I will not be recording the class sessions because it's not going to be an easy thing for me to do. But as always, I will be recording these lecture videos and posting them to Blackboard. No new material will be introduced during the review sessions. It is perfectly legitimate for you to remain online. You just have to make sure you're keeping up with these videos. You will have a quiz next Friday. That is September the 18th, I believe. And that quiz is going to cover chapters 8 and 9, okay? Chapter 8 is on enzymes. Chapter 9 will be on cellular respiration. The week after that, so what would that be? Let's see here. That would be the 25th, maybe? I'll have to double check that. You will have an exam. That exam is going to cover Chapter 7, Part 2, all the way through Chapter 11. And if you're following along on your syllabus, this is exactly what is posted on the syllabus, okay? So all of this is on the syllabus, as they say. All right, so that's the housekeeping stuff taken care of. Let's just kind of back up for a second and remind ourselves where we were. We were talking about enzymes, and I mentioned to you that enzymatic reactions proceed in 3 phases. You have the initiation phase, which begins with the binding of substrates to the active sites. Then you have what's called the transition state facilitation. So this is the middle part of the enzymatic reaction. And what's happening here is a little bit of an induced fit whereby the enzyme begins to kind of clamp down on and force these substrates together so that we can break old bonds and form new ones. So here you can see that this bond between B and C is being stretched, and it's bringing this little molecule here into closer proximity with A. So here we're going to start forming a new, this is B? Yeah, B. Here we're going to start forming a new bond, and we're going to break that bond, okay? And then the last phase of an enzymatic reaction is called termination. Once the bonds have been shifted, that means that these products no longer fit in the active sites like they should, okay? So in other words, we say that they have a lower affinity for the active site and they are therefore released, okay? So that's where we left off. Today, I want to talk about things that affect enzymatic reactions. In other words, what determines how fast these reactions occur? I told you before that the purpose of an enzyme is to speed up the rate of chemical reactions, that without enzymes, chemical reactions would occur too slowly to sustain life. So the whole point is for them to act rapidly. So what determines how fast an enzyme can make a chemical reaction go? Well, there are several factors. All right. The first one I kind of want to demonstrate by showing you this graphic here. So on this particular figure, what we have is on the x-axis, we have the substrate concentration. And remember, the substrate is just the reactants in an enzymatic reaction, okay? Reactants. But we call them substrates. And so on the exam and on quizzes, I will be referring to them as substrates. So we have the concentration of substrates. And then here we have the rate of product formation. So this is really equal to the rate of the enzymatic reaction, how rapidly it's turning those substrates into products, okay? So this is equivalent to the rate of the reaction. So let's take a look at what's going on here. Let's look at this blue line first. This is an example of what happens without an enzyme. So uncatalyzed reaction, no enzyme. And what you would find is that without enzymes, if you increase the reactants or the substrates, if you increase their concentration, you increase the rate of the reaction, okay? But remember that the entire job of an enzyme is to speed up chemical reactions. So let's look what happens in the presence of an enzyme. This red line is a catalyzed reaction. So that means we're using an enzyme to form the product. And look here, as we increase the substrate concentration, the rate of product formation, which is equal to the rate of the reaction, starts going up like crazy, crazy, crazy. It goes up so rapidly until it tops off. It kind of maxes out up in here, okay? So you can see that enzymes very, very rapidly increase reaction rate. But there's a point at which the ability of enzymes to catalyze chemical reactions sort of tops off. Why is that? Well, let's just pretend that you have 20 enzymes, okay? That means you have 20 active sites, right? So if you only have one set of substrates, it can't go very fast. 2, it increases. 3, 4, 5, 6, 7, 8, 9, 10. As you start increasing the amount of substrates up to 20, you increase how quickly you can turn those substrates into product. But at some point, let's say that you had 40 sets of reactants or substrates. Again, I'm using these interchangeably. So let's say that you have 40 sets of substrates, but only 20 enzymes. It's only so fast that 20 enzymes can work. At some point, you run out of active site parking spots for the substrates when you get to high concentrations. So what I have done here is when the enzyme concentration is held constant, adding more substrate increases reaction rate until all enzymes become saturated. Or another way of saying that is all the enzyme parking spots are full. So this point right here represents enzyme saturation. Okay. So in other words, all of the active site parking spots are full. So two things that can affect how fast enzymatic reactions occur. One is substrate concentration. And I'm going to abbreviate that by writing the word substrate with brackets around it, because when you take chemistry, you'll learn that these brackets are shorthand for concentration. The second thing that can affect these enzymatic reactions are the enzyme concentrations. Okay, so if we, let's just say we had 40 substrate sets and only 20 enzymes. Well, we've topped out, right? We've occupied all our parking spots. But what if we increased the number of enzymes to 60 enzymes? Can you see that you would speed that reaction back up? okay? So two things that can affect how fast these enzymatic reactions can go. One is substrate concentration, and the second is enzyme concentration. There are more things that can affect how rapidly these reactions can proceed. I want you to think back to chapter 3 when we talked about proteins, right? And I told you, first of all, that enzymes are proteins, right? Enzymes are one type of protein. And you know that proteins have a very particular shape in their tertiary structure, right? Where they have a three-dimensional, knotted up shape. And that sometimes you can disrupt this three-dimensional shape in a process known as denaturing, where you cause the protein to unfold, right? And if you remember, two factors that could denature proteins were temperature and the amount of acid, or the pH of the solution that things were sitting in. So two more things that can affect the rate of an enzymatic reaction are temperature and pH. These graphs are showing you examples of enzymatic reactions from two different bacterial sets. And what these bacteria are doing is they're digesting chitin. Okay, so they're digesting chitin. And in your book, it tells you that these are reaction rates for an enzyme called chitinase. And chitinase just breaks down chitin. Chitin is the primary component of exoskeletons on insects and arthropods, and it's also in the cell walls of fungi. So these are bacteria that are able to digest chitin, and they have this enzyme chitinase. Bacteria that live in a cool and neutral environment, if you look at how rapidly their enzymatic reactions occur, you can see that at low temperatures, because molecular motion is really low, you don't have a lot of molecules moving about, the reaction rate is pretty low. As we increase the temperature, the reaction rate increases until we reach what's called the optimal temperature for that enzyme. At that point, the reaction rate starts to rapidly decrease because the enzyme starts to unfold or denature. And I'm just going to abbreviate, okay? Here we have the same enzyme, but from a different bacteria that lives in a hot and acidic environment. And if we look, we see a similar trend. We see that at low temperatures, the reaction rate is slow. This is probably due to decreased molecular motion. And then we reach a point that's what's called the optimal temperature for that enzymatic reaction, where it's moving at its fastest rate. And then once you hit that optimal temperature, if you exceed that, you see that the reaction rate begins to dramatically decrease because that enzyme denatures or unfolds. Now, what's the difference here and here? Well, remember us talking a little bit about the fact that proteins are made up of amino acids, and amino acids have R side chains that have different chemical compositions. So what's happened is the bacteria that live in a hot and acidic environment have a few changes to their chitinase enzyme, where they've added some amino acids with R groups that are more tolerant of high temperatures than these guys, okay? And here we have a similar situation with pH. In this example, we have chitinase from bacteria that live in a hot and acidic environment. So remember, low pH equals acid, high pH equals base. And so here we can see that at very high acid concentrations, this enzymatic reaction is proceeding quite slowly. We have a lot of protons. Here we have an optimal pH, and then it begins to decrease. And again, this is due to the enzyme unfolding and not being its proper shape outside of its optimal range. We see the same thing here with our cold and neutral bacteria, okay? And again, these differences in the chitinase activity are due to differences in their amino acid sequence, where this guy has substituted some amino acids that have R side chains that are more suited to a hot and an acidic environment. So chemically, they're better in those environments, while this guy has some R side chains that are better suited to cool and neutral environments, okay? So temperature and pH can also affect enzyme functions, all right? What else? Other regulators of enzyme activity, sometimes enzymes do not work alone. They need helper molecules in order to carry out the enzymatic reaction. And three different types of helper or partner molecules that we might encounter are cofactors, coenzymes, and the last one is something called a prosthetic group. Okay, so an enzyme cofactor is often an inorganic, well, it is an inorganic molecule. So something like zinc, which is abbreviated ZN, and it usually is found in ionic form in your body. Magnesium is another example of a cofactor. It is also found in ionic form in your body. Iron. Okay, these are all examples of cofactors. So what do these guys do? Well, what often happens is that an enzyme by itself, the active site might not be just the exact right shape, but these cofactors tuck into the active site and make it a more suitable place for the substrate. Here, I've made it a better shape, but it might also make a better charge, okay? So these cofactors are able to be added into the enzyme in order to make it a better fit for the substrate, okay? So we'll just put better fit. Better fit for the substrate, okay? Incidentally, these probably look familiar to you. You probably recognize these as things that you find in vitamins, right? In multivitamins. Many of the multivitamins that we take simply because they are enzyme cofactors. And if you are deficient in these vitamins, then your enzymes don't work properly, and that can have health effects. So I'll give you an example. Vitamin C vitamin C is an enzymatic cofactor. Okay. It works with one of the enzymes that helps to produce a protein called collagen. Collagen, I think I've told you before, is a structural protein. It's part of the extracellular matrix, and it does things like it provides structure to different tissue types like your gums and other things like that. If you lack vitamin C, you start having problems like bleeding gums and poor teeth and things like that. And that is a condition that you may have heard of, like in pirate novels, called scurvy. So scurvy, like, that's a B. Scurvy is a condition where you have like bleeding gums and bad teeth and bad skin, and it's all because you lack vitamin C cofactors to make collagen. And it was common on sea voyages because if you were at sea for a long time, you didn't have any fresh fruits or vegetables to supply vitamin C for you. So if you've ever like seen a like old pirate movie like Pirates of the Caribbean or something, and they're like, you scurvy something or other. Well, scurvy was common among pirates because they didn't get fresh produce. All right, moving on. Coenzymes work almost exactly like cofactors. The difference is that these are inorganic molecules. So these do not contain carbon. This is zinc, this is magnesium, this is iron, okay? They don't contain carbon. Coenzymes are organic molecules. They're larger, okay? In other words, they are carbon, maybe carbon ring kind of molecules, okay? But they work essentially the same. And we're gonna see examples of coenzymes in our next chapter as we begin to study cellular respiration, okay? But essentially they work the same way. If we go down here, You can see this says cofactor or coenzyme. It's working the same way. It's basically altering the active site to make it a more suitable place for the substrate. The last type of helper molecule is something called a prosthetic group, okay? So these are non-amino acid molecules. I'm going to put non-AA molecules that are permanently added to a protein in order for it to function. So this is a permanent addition, okay? By contrast, these are not permanent, okay? These can bind and unbind. This is permanent. A great example of a prosthetic group, you've probably heard of hemoglobin. Hemoglobin is made up of 4 independent protein subunits. So this is an example of a protein with what's called a quaternary structure, okay? And if you don't understand that, go back and look that up in chapter 3. But in order for hemoglobin to function, it has to have a very special chemical group called a heme added to it, all right? This is a heme group here, here, and here. This is actually what oxygen binds to so that hemoglobin can carry oxygen, okay? So if without these heme groups, this particular protein or enzyme would not be able to do its job. So that's a great example of a prosthetic group, all right? Now, there are other ways that enzyme activity can be regulated. Sometimes you'll have a molecule that looks like a substrate. So I'm going to put molecule that looks like a substrate. Okay, so that means that it can bind to the active site of a particular enzyme. And if this imitation or imposter molecule is bound to the active site here because it kind of fits, then the real reactants, the real substrates, aren't able to bind and you get no product, right? So let's take a look again. This is the real set of substrates. Okay, this is what really belongs in these active sites. But this is a molecule that kind of looks like these substrates, and it's able to weasel its way in to these active sites and block the substrates from getting to their parking spots. This effectively blocks the enzymatic reaction and means that no product can be formed. This is called competitive inhibition. And I'm going to abbreviate because it's written right there. I'm just giving you time to write. Now, why would that be? Because this molecule is competing with this molecule for the active site, isn't it? So let's think about what that means. The more of these guys there are, the less product can be made, all right? So let's think of an example. Down here, I have the example of cyanide. Cyanide acts as a competitive inhibitor for a particular enzyme called cytochrome C oxidase. This blocks the electron transport chain, meaning the cell can no longer produce ATP for energy. If that is a meaningful example for you, go for it. I have a more timely example for you. You may have heard of something in the news called remdesivir. I may be misspelling it. I'll look it up. This is an antiviral drug that was initially thought to help with the treatment of COVID-19. And I haven't looked at the data lately. I think it's sort of a mild, and maybe it was maybe a little overhyped when we first started talking about it. But why did it work? Why might this have worked? Well, it's because it used competitive inhibition. So COVID-19 is a virus, right? Viruses get in your cells and they trick your cells into making more virus, right? That's what they do. And the way they do that basically is they get the cell to make copies of viral DNA. So what's remdesivir? It's a fake nucleotide. We call it a nucleotide analog. So it basically functions by competitive inhibition. These fake nucleotides get into the enzymes that are making this viral DNA, and therefore the proper product is not formed. The viral DNA doesn't get made because instead this fake nucleotide is parking in the parking spot, okay? So that is an example of competitive inhibition. So in addition to competitive inhibition, there are other ways that we can regulate enzyme activity. And in this example, we're going to talk about something called allosteric regulation. Okay, now let's just think for a second about this word, allosteric. The term steric in chemistry usually has to do with the shape of a molecule, okay? And the prefix allo usually means other, at least in the chemistry context, okay? So allosteric regulation is going to be involved in creating different shapes for our enzymes other than how they would ordinarily exist in the cell. So let's first talk about allosteric regulation in terms of activating an enzyme. So in the absence of regulation, you've got this basic shape of this enzyme, and hopefully you can see that these active sites, can you kind of make out that they're oblong, and they're just not the precise shape or orientation for these substrates to fit in here, right? But if we use an allosteric regulator, okay, so an allosteric regulator is a molecule that changes the shape of the enzyme. So let's write that down. Allosteric regulator. Regulator, and I'm just going to put reg. It changes, and I'm going to use the delta symbol for change, changes the shape of the enzyme, okay? So in this activation example, this regulatory molecule, this allosteric regulator, binds, and it causes the shape of this enzyme to change so that the active sites are now perfectly suited for our substrates to bind, okay? So this is allosteric regulation as it pertains to activating an enzyme. But we can also use allosteric regulation to inhibit an enzyme. So in this instance, it's just the opposite of what we saw here, where when the enzyme is by itself without the regulatory molecule, the active site is perfect and substrates can bind. But if the regulatory molecule binds, then it alters the shape of the enzyme, makes these active sites inappropriate for the substrates to bind to, and we've stopped the enzymatic reaction. So allosteric regulation is when the regulatory molecule causes a conformational change. Remember, these are proteins, so these are conformational changes, making the active site available to the substrate, okay? In inhibitors, like here's an example of an inhibitor. Let's see here. Allosteric inhibitors of thrombin have been discovered, which could be potentially used as anticoagulants. So, you know, I didn't plan on talking about, so you can ignore that. That was from a previous semester, all right? But in general, just understand how these allosteric regulators work. All right, so let's change gears. So up until now, we've been talking about how we can regulate enzymatic reactions. So let's just summarize that. I'm going to throw a new slide in here really quickly. Let's just get rid of these. And let's just list some of the ways that we can regulate how rapidly an enzymatic reaction proceeds or whether it proceeds at all. Okay, we can alter the enzyme or the substrate concentration. So I'm going to enzyme substrate concentration. Okay. We can change the temperature or the pH of the environment, and that would change how the enzymatic reaction proceeded. The availability of cofactors, coenzymes, availability of cofactors, which were those inorganic molecules like vitamins, or coenzymes, which were larger carbon-bearing molecules. The availability of these guys can determine whether or not an enzymatic reaction proceeds. And then we can use regulators in the form of competitive inhibitors, in the form of allosteric regulations, which can either turn on or off the enzymatic reactions. If these don't make sense to you, you need to go back through this series of slides, okay? I definitely ask questions about this stuff on my quizzes and exams. All right, but let's switch gears for a second and let's talk about linking the action of enzymes together to begin to form something called metabolic pathways. So oftentimes you can have a situation where you have enzyme 1 and it makes product A. Product A will often be the substrate for enzyme 2, which converts product A into product B. Product B is often the substrate for enzyme 3, etc. So you can see that we can link these three things together in order to make a particular product. And this series of enzymes would represent a metabolic pathway. Now let me give you a real world example of this that maybe will help this make more sense for you. I'm gonna do a little erasing here. What I have for you is some examples of real world metabolic pathways that make some of your hormones. Aldosterone is a hormone that's involved in regulating your blood pressure. Cortisol is your stress hormone. It is involved in not so much fight and flight because that's epinephrine, but it's involved in like when you get anxious because you're stressed out, that's usually cortisol in action. And then testosterone, and this is the European spelling for estrogen or estradiol. Okay, these are of course your sex hormones, right? So in this example, we have the beginning molecule, which is cholesterol. And there's an enzyme that converts cholesterol into this product here, pregnenolone, okay? You don't have to know these words. I'm just showing you this as an example, okay? This enzymatic product, here we go. This product gets converted by this enzyme to this product. This product gets converted by this enzyme to this enzyme to this enzyme to this, and you end up with your final product, okay? And the neat thing is you can have branches off of here. So here we've got this product can be converted by this enzyme to this, by this enzyme to this, and then you can follow this pathway. This can be this enzyme to this. You can see where I'm headed here is that each of these represent links in a chain to end up at these final products. So these are examples of metabolic pathways to make these various hormones, okay? And again, please don't study these specifics. I'm not going to ask you these specifics. I will ask you to understand what a metabolic pathway is. And it's simply when you link enzymes together so that the product of one enzyme becomes the substrate for the next one, okay? Product, substrate. All right. Now, in thinking about metabolic pathways, a couple of vocabulary words that I do want you to know, I'm going to erase this here, so I still have some room to write, are the terms catabolic and anabolic. All right. Anabolic pathways build molecules, okay? Whereas catabolic pathways break them down. So in my example here, this is an example of an anabolic pathway because we're building a molecule. We're taking cholesterol and then we're doing different things to it to build a molecule here, aldosterone, which is a blood pressure regulating hormone. But if I were going to break aldosterone apart using a series of enzymes to recycle this, then that would be a catabolic pathway, okay? All right. Now, metabolic pathways can also be regulated, okay? So we regulated individual enzymatic reactions using these mechanisms, but we can also regulate an entire pathway of enzymes here, and often we do that using something called a feedback mechanism. And in particular, what I want you to understand is something called negative feedback. Okay. In negative feedback, you have a situation where you've got a substrate and it's going to enzyme 1. And then you have product A that's now the substrate for enzyme 2. Product B is the substrate for enzyme 3. At some point, you might in your body have made sufficient final product that you don't need to make anymore. And so when that occurs, what happens is the concentration of this product builds up enough that it can come along and bind to enzyme 1 to shut the whole thing down. And the way it's doing this, if you look, we've got an allosteric binding site here. That's going to induce a conformational change so that the active site is no longer the right shape or orientation for this initial substrate to bind. So this whole pathway gets shut down. And the way it gets shut down is by this final product building up to sufficient concentrations that it binds to this active, or to this allosteric regulation site. Let me give you a real world example of that. These are 2 amino acids. One is called threonine and one is called isoleucine, okay? You need both of these in order to build proteins. And it turns out that isoleucine is made from threonine, okay, through a series of enzymatic reactions. But eventually your body has enough isoleucine, and we don't want to keep converting this amino acid into this one if we don't have to. First of all, it probably consumes energy in the cell. And second of all, we don't want to deplete this supply to virtually nothing either. So what happens is as the concentration of isoleucine product begins to build up, it starts to bind to a regulation site on this first enzyme, which will then shut this whole thing down. Okay, that is an example of negative feedback because what you have is feedback of the product on the metabolic pathway, and it's negative because it's turning it off. All right, Positive feedback also exists, and it's a little bit more complex. And so we may or may not get around to talking about that this semester. All right. The last thing that I want to talk to you all about is the kinds of chemical reactions. So we're switching gears entirely now. We're moving from enzymes themselves to talking about types of reactions that enzymes might catalyze. So this is going to be types of chemical reactions. And specifically today, we're not going to go through all the various types, but there's one very important type that I want to talk to you about because it is going to be the subject of chapter 9 on cellular respiration and chapter 10 on photosynthesis. And these chemical reactions that I want you to know about are called oxidation reduction reactions. They are often abbreviated redox reactions. Now, what's happening in an oxidation reduction reaction? Okay, if something is being oxidized, it is losing electrons. Now, let's think about this. And I'm going to put E negative for an electron. This should make sense to you because we know that oxygen is an electron scavenger. It likes to rip electrons off of something. So if something is being oxidized, that means that you can kind of think of it as having been attacked by oxygen and losing an electron. If something is reduced, it has gained an electron. Now you may be thinking, well, if it's gained something, how is it reduced? Well, what it's gained is it's gained charge, okay? It's gained, sorry, it's reduced charge, sorry. What it's reduced is it's reduced its charge. It's reduced its chemical charge. And that's where that name initially came from, but it's also been a source of confusion for students for many, many moons, okay? So oxidation is when you lose an electron, reduction is when you gain an electron. Where this terminology comes in is you have a reduced charge. But there's a great mnemonic device that everyone uses to remember an oxidation reduction reaction. Oil rig. Oxidation is loss, reduction is gain, okay? Oil rig, oxidation is loss, reduction is gain. And I think I have that for you in your notes here. Yeah, oxidation is loss, reduction is gain, okay? Why is this important? Well, it turns out that oxidation gives off energy while reduction absorbs energy, okay? In other words, oxidation is exergonic and reduction is endergonic. Okay, so that's what this is here. It's showing you, remember how I showed you how we can use an exergonic reaction to drive an endergonic reaction. And let me give you an example that's going to lead us right into our next chapter. All right, so in this example, we are going to be oxidizing glucose, okay? Which means that glucose is going to lose electrons, right? So let's follow it over here. This molecule that was initially glucose, here it's been oxidized into carbon dioxide, okay? So if we take glucose and oxygen together, we oxidize glucose into carbon dioxide and we reduce oxygen into water, and this gives off energy. Taking glucose and adding oxygen to create carbon dioxide and water is the definition of cellular respiration. And that is the subject of respiration That is the subject of our next chapter. So this oxidation reduction reaction is going to give off energy, and we're going to use the energy of this reaction to make ATP. Now, the reason that I stuck this in your enzyme lecture is it is in this chapter, but it also, all of this is happening, these chemical reactions are happening because enzymes are catalyzing these reactions. So enzymes are causing this to occur, and this is a preview of where we are headed, okay? And that, I think we're going to eliminate this slide from this lecture here, and that's where we're going to end chapter 8 for today. So let me know if you have any questions, and I'll talk to you soon. Bye-bye.